Hello, everyone. We are going to be going through questions 11 through 15 in the life sciences practice exam for this video. So we're going to start out with question 11. So it says the major source of energy for cellular respiration in plants is provided by which of the following? A, sunlight, B, carbon dioxide and water, CO2, carbon dioxide, C, nutrients absorbed from the soil, or D, sugars synthesized from photosynthesis. So the key word here is cellular respiration. If you immediately went to sunlight, you're thinking of photosynthesis, which is important in plants, but this specific question is talking about cellular respiration. So make sure we're focusing in on what they're asking for. So let's start with the wrong answers. So A, we just discussed sunlight is an energy source, but that's in photosynthesis, not cellular respiration. So it's not A. B is incorrect because carbon dioxide and water are used. They're important, but they're not energy sources. So they're part of the process, but they're not a source of energy. And then C is incorrect because, again, nutrients are important, but they're not the energy source here. So in this case, D is going to be the correct answer. So D says sugars synthesized by photosynthesis. So D is correct because those sugars that are produced in photosynthesis are then put back in for cellular respiration to be used as energy to fuel that process. I'm going to pull up a picture here so you can kind of see how these two work together. So photosynthesis is up here. That's going to happen in the chloroplast. So you're going to have water and carbon dioxide going in. Those are your reactants in photosynthesis. And then your products of photosynthesis are going to be O2 and glucose here. And so the O2 and glucose are then going to go into cellular respiration. So these are our reactants in cellular respiration to create water and carbon dioxide, which are our products of cellular respiration. And you can see here, just as a refresher, photosynthesis is the chloroplast, cellular respiration here, it's going to happen in the mitochondria. So both important processes, but this specific question is asking about cellular respiration. So the answer is going to be D, those sugars synthesized by photosynthesis. Let's go back and look here now at question 12. So question 12 says, in eukaryotes, which of the following molecules is produced in anaerobic cellular respiration, but not in cellular respiration? So this question is kind of one that you kind of just have to memorize, anaerobic versus aerobic cellular respiration. So I've got a diagram and I've got just a comparison chart, whichever kind of person you are. But so here you can see on the left, we have a picture of aerobic respiration. This basically means you can see aerobic means with oxygen. So you can see oxygen is being put in here. And you can see we're able to make quite a bit of ATP. That's our energy sources. So we've got 2, 4 plus 32. So we've got 36 ATP being made here. In aerobic cellular respiration. This is the one, if you're just going out for a walk, you've got lots of oxygen. This is the process that you're going to be able to use. You're going to use aerobic respiration because you have oxygen. Once you run out of oxygen, so all that oxygen is used up, we have to move into this process called anaerobic cellular respiration. So this is where you can see there's no oxygen being put in here. So you can see we've got glucose. This is going to go into more detail on the specific process if you want to look more into it. But basically, you can see here at the end, we're creating this lactic acid here. This is what makes your muscles sore or makes you get tired, is that lactic acid builds up. You're going to get tired and things like that. So here's a good kind of overall picture. If you're a picture person, if you want to pause the video here and write anything down or take notes over that. And then I'm going to scroll down. Here is a or just a chart if you're a chart person. So you can see the reactants in anaerobic glucose, only glucose goes in. Aerobic, we said, uses oxygen, so the oxygen is going in. Combustion and complete versus complete. Energy yield, again, you can see here, aerobic is a lot higher. It's going to produce 36 to 38 ATP, more energy, versus anaerobic don't have a whole lot, only 2. Products, again, here you can see aerobic lactic acid for us animals, yeast, ethanol, carbon dioxide. You can see the products here. Location, cytoplasm versus aerobic is the cytoplasm and it's the mitochondria. The different stages here. Again, you saw that in the picture as well. So now let's go back and look at the question again. So this is question 12 that we're working on here. So using these diagrams, you can see that the only thing, sorry, that lactic acid is created in anaerobic respiration, but not aerobic respiration. Lactic acid is what makes your muscles weak when you exercise. We talked about this. So when you run out of oxygen, your body is going to switch from aerobic to anaerobic respiration. So the only question here, or the only answer choice that has to do with anaerobic cellular respiration, but not aerobic respiration, is going to be lactic acid, A. So we saw that that only happens in anaerobic respiration. So let's go down now to question 13. It says, which of the following is most likely to trigger uncontrolled cell division in tumor formation? So a mutation of the cyclin protein that interferes with the binding of cyclins and cyclin-dependent kinases. B, a mutation to a cell surface receptor that causes continuous signaling in the absence of ligand binding. C, an increase in the cytosolic concentration of second messengers such as CCAMP and calcium. Or D, increase in the rate of cellular respiration and ATP production. So again, let's go through the wrong answers here. So A is going to be incorrect because the blocking interaction between cyclins and cyclin-dependent kinases, it's going to block the cell cycle and it's going to block that cell division. So it's not going to continue to happen. So those are important for cell division. So again, if we block those, it's going to keep it from dividing. It's not going to make it divide too much, which is what we're looking for. We're looking for continuous division. C is also incorrect because CCAMP and calcium are ubiquitous second messengers. So that means increasing their concentrations is likely to cause the metabolic problems that a cell has, and it'll cause it to die. So again, not increasing, it's going to decrease because it dies. If we up these two, then again, we're going to stop dividing, not divide too much. So not C. D is incorrect because increased cellular respiration and ATP production are likely to be consequences of rather than cause the increase of cell division. So we would get more of these cells dividing, but this would not cause the cell to divide more. So in this case, B is going to be correct because the binding of ligands such as cellular growth factors to the cell surface receptors is the normal way by which signaling is triggered. So if we have a mutation to those receptors, it's going to cause it to induce conformational changes to those receptors, causing those receptors to initiate signals even when we're not binding. So it's going to think that something is bound. It's going to say, hey, keep going, keep going, keep going. Even if something's not there, it's going to continue to replicate when it shouldn't. Sometimes if you don't know the answer, you can just look for trigger words. So in this case, this answer says continuous. So usually when we're talking about cancers and tumors and things like that, the word continuous is going to be kind of a trigger word. So even if you weren't sure, if you saw that word continuous, that would be a potentially helpful word to kind of look for with cancers and tumors questions. All right, so let's look at 14. 14 says the drug used to treat some type of cancers are toxic to rapidly dividing cells. Which of the following best explains why patients whose bone marrow has been damaged by such drugs often have to receive a bone marrow transplant? So we're going to switch it up here. We're going to actually talk about the correct answer first. So the point of this question is that the drug targets rapidly dividing cells. So your cancer cells are rapidly dividing, but there are also other cells in your body that are rapidly dividing. So the question here is basically asking you to recall that bone marrow is made of rapidly dividing cells, which is answer choice B. Bone marrow contains stem cells that give rise to red and white blood cells of an individual. So if we look at, let's go through the wrong answers also just to knock those out. So A is incorrect because it's a true statement, but it doesn't have anything to do with rapidly dividing cells. So A says bone marrow must be dense so that the bones can support the mass of an individual. So again, true statement, but nothing to do with rapidly dividing cells. C is incorrect here. C says bone marrow contains adipose cells that store fat as a source of energy for an individual. So C is incorrect because bone marrows do not store a significant amount of adipose tissue, providing an insignificant amount of energy. And also, again, it has nothing to do with rapidly dividing cells. D is incorrect because while calcium is in the bones, it's not in the bone marrow itself. So again, also not related to rapidly dividing cells. And I realized we never read the answer choices. So B is the correct answer. It says bone marrow contains stem cells that gives rise to red and white blood cells. So again, knowing that red and white blood cells that are made in the bone marrow are rapidly dividing cells is what they're really targeting us here with this question. All right, so question 15 says if a single base pair is deleted from the coding region of gene X, which of the following describes a possible effect of the deletion of protein X? Sorry, of the deletion on protein X. So again, a single base pair is being deleted. So A says protein X is going to be normal because the sequential pairs must be deleted to affect the protein sequence. B, protein X is going to be normal because every 3 base pairs is a wobble base that can be deleted without altering the protein sequence. C, protein X is going to differ from the normal sequence by the single amino acid coded in the region of a deletion. Or D, protein X will be significantly shorter than the normal length because a stop codon will be introduced downstream of the deletion. So before we go through the answers, I want to do an overview of mutations. So if you got this question right, you understand this, you can skip this question. But if you didn't get this question right, we're going to do an overview of mutations here. So I'm going to pull up a picture. Here we go. So I thought I had another picture here. So yes, okay, I guess it's this picture. So this is going to show us the examples of different mutations here. So before we go through this, a quick refresher. We start with DNA, and then that DNA is going to go to, so we can see that here. Here's our DNA. And then after we have DNA, we're going to go to mRNA, which is the second row. And then after mRNA, we're going to go to proteins. So the DNA here is going to be used as a template to create the mRNA. When we're going from DNA to mRNA, it's important to remember that A pairs to U, and you can see that in this normal one here. Whenever we have an A, it's going to a U below it. And then whenever we have a G or C, it's going to pair to G or C. So we can see here we have a G, it's pairing to a C on the mRNA, or here we have a C, that's pairing to a G. So A will always go to U, and G will always go to C. Now you'll notice we have T's in our DNA. There are no T's in RNA. So if we have a T on the DNA, that's going to go to an A here on the mRNA. And again, these letters are different nucleic acids: adenine, guanine, thymine, cytosine, and uracil. So these are all the nucleic acids. So then that mRNA is then going to be used to turn into protein. So I'm going to scroll us down, or I guess scroll us up and go back and look at this other picture here. This is called a codon chart. Hopefully you have seen one of these before. But basically you're going to start in the middle with your first letter, and then you're going to move out to the second circle here for our second letter. And then we're going to go out to our third row here for our third letter. And so as we go, let's say we had UUA, we would go U, U, A, and that would tell us that the amino acid that's going to be created here is going to be leucine. Or let's say we had AUG. We'd go A, we'd go to U, and then we'd go to G, and that's methionine, you can see here, and that is our start codon. So every time it's going to go through and it's going to scan the amino, or it's going to scan the mRNA, and it's going to look for that AUG. And as soon as it finds that AUG, that's when it's going to start coding. And then you can see here, these squares represent our stop codons. There are multiple different stop codons that we could have here. So as soon as it finds that sequence, so for example, UAA, that's going to stop and it's going to terminate that protein sequence right there. So let's go back again and look at this picture. So here we have our normal sequence. This is what it's supposed to look like. Then if we scroll down, the next one we have this. These are all considered point mutations. So a point mutation, that means one single base pair is affected. So in this case, we have a substitution. So if we look at our normal, looks like in our normal, we have a C right here in the third codon, right here. Can I mark? Okay, yeah. Let's just mark. So in our normal, we have a C right here. Now in our substitution, you can see it's been substituted for a T. So you can see how it changes. So C will go to G. If we change that to a T, T here is now going to A. And you can see GLU turns into LYS, to lysine down here. So that one single base made a change. However, you'll notice everything else stays the same. All the rest of them are all the same. So it's not a huge error, because for the most part it stays the same. So now let's scroll down and let's look at, see, let me erase these that I did. Okay, so now we're going to scroll down and we're going to look at insertion here. Annotating feature back. Okay, so now instead of substituting, we're going to add in an extra base. So here you can see we have CTT in our normal. Here's our normal, CTT. Now they're adding in, here's our original CCT. Now they're adding in this extra C. So they've inserted that. So again, you can see how that changes your mRNA. Instead of GAA, we have GGAA. And then you can see the ones in front stay the same. We still have phenolphthalein and we still have tyrosine. But now we can see we have a change here in glycine and we have a change here in arginine. So before, in the one above with the substitution, you can see we only had one amino acid change. But when we insert, everything shifts down. So your entire, we call it a frame, your reading frame changes because we read in sets of 3. So when we insert, all of a sudden, every single set of 3 is going to be changed. So that makes a huge difference here. Instead of just changing one nucleotide, one amino acid, we're changing every single amino acid that comes after it. Erase these, and then we'll look at our last one. So let's now scroll down and look at deletion. So when this is the one that our question is talking about. So again, here, if we go back up, oh, I wish I could make this all, maybe I can make this, let me see if I make this frame bigger, if we can get, I don't think we're able to get them. Hide that maybe. Okay, kind of, they're kind of on the same frame now, at least we can see the DNA. So here we can see we have A, in the normal, we have AAA. Get my annotating back. AAA is the same. And then here we should have a fourth A, or the start of our next reading frame starts with an A. Here you can see on deletion, they're deleting that. They're taking that out. So now instead of that second codon reading, or that second reading frame reading ATG, now it's reading TGC. So you can see everything is shifting up this time. So every single reading frame, therefore every single codon in the mRNA and every single amino acid is going to be changed. So I'm going to scroll down. We're going to kind of lose. Let's get exit out of that. Erase my. So again, we are normal. You can see up here we started out with these. Our first one stays the same because it happened before the deletion, but now you can see every single amino acid after the change, after the deletion here, is affected. So substitution, it's not a great mutation. Obviously, it still changes something, but it only changes one amino acid here. It only is going to change this one. However, insertions and deletions, we change lots of everything after that insertion or everything after that deletion gets changed. So that those are usually a lot bigger deals than a substitution. And again, these are all called point mutations. There's other types of mutations that affect, there's chromosomal mutations where entire chromosomes are affected. But in this case, we're just looking at point mutations for this question. So let's go back and look at the question now. Again, and let's reread it. So it says if a single base pair is deleted, so again, we're looking at one single nucleotide base, is deleted from coding region X, which of the following describes a possible effect of the deletion on the protein? Sorry. Okay. So right off the bat, so if we have a deletion, all the nucleotide bases, again, are going to shift up one, which means all the amino acids are going to change. So the protein is definitely not going to be normal. So we can eliminate A and B. So now we know it's either C or D. So just as we said above, with a nucleotide deletion, all of them are going to shift up, causing all of those amino acids to change. So we can eliminate C. Again, C says protein will differ from the normal sequence by a single amino acid coded in the region of the deletion. So this means that only one single amino acid is affected. And if we go back and look at our pictures, you could see with a deletion, we had everything behind it was affected. So it's not just one. So that eliminates C because it's not a single amino acid. So in this case, that's going to leave us with D. Protein X will be significantly shorter than the normal sequence because a stop codon will be introduced downstream of the deletion. So again, the question here says which is possible. So this is a possible thing that could happen. It could shorten the protein by introducing a premature stop codon. However, again, this is more than the question is asking, but I just want to point this out. It is possible that it could be longer than normal. It could take longer for another stop codon to randomly appear. But again, this question just says possible. So again, we know it's not A and B because we know the protein's not going to be normal. And we can eliminate C because it says a single amino acid change. And we know that it's going to be everything after that. So it's going to be a lot more than that. So that is all for questions 1 through 15. If you have other questions on other questions for this video or for this test, you can go ahead and find that next video that's going to relate to your questions.